Hello, IB Bio students. Today we continue with 11.4 and we are looking at the structure of the egg and sperm. So this is a skill where you're going to have to be able to annotate a diagram of a mature sperm and egg. So you might be given something like this without the labels, and you will have to include the labels of the structures that you're looking at and also include an annotation, meaning what is that particular structure responsible for. So let's start with some general information here. So we know that male and female reproductive gametes are called sperm and egg. And as always, form fits function, right? So the male sperm is small and it's motile, meaning it can move, it can swim. And we know that the sperm only contributes the male's haploid nucleus to the zygote and perhaps the centrioles, and that's about it. Whereas the female gamete, or the egg, it's very large. We notice the distinction between the primary and the secondary oocytes and the egg at the end of reproduction. And we see that the reason why it's so large, it's because it contributes all the organelles and cytoplasm to the zygote. Now, the female gamete is non-motile, meaning that it cannot move by itself. It just kind of flows via the cytoplasm and cilia movement within the oviduct, and it allows the egg to pretty much just passively move from the oviduct down the fallopian tubes. All right, so let's start looking at the structure of the sperm. So the sperm, or the spermatozoa, is divided into three main sections. We have the head, we have the midpiece, and we have the tail, or also known as the flagella. So what's inside the head? So inside the head, we have the acrosome, and this is kind of like a little cap here, and it's full of hydrolytic enzymes, which means that enzymes in here are going to penetrate the jelly coat here in the female's egg. Then we have the nucleus, and we know that it's a haploid nucleus with half the number of chromosomes from the male parent. And then we have centrioles here that are going to be needed by the zygote when the zygote is about to divide after fertilization, because we know that the ovum expels its centrioles. The midpiece is filled with mitochondria, and we know that mitochondria provide energy, and we know that the sperm is motile and needs to swim, therefore it gets its energy from the mitochondria. And then the tail, or the flagella, is composed of a microtubule structure called an axoneme, and this is what is going to allow for a wide range of motion of the tail. Now, the egg, when we look at the structure of the egg, we recognize that it has two distinct layers. So we have the zona pellucida, which is a jelly coat here, that needs to get penetrated by the enzymes that are found in the acrosome of the sperm. And then surrounding that, we have the corona radiata, which are just follicular cells, and these follicular cells are going to provide support and nourishment for that egg cell. Now, inside the egg cell, there are other organelles that we don't see here, along with the cytoplasm. And we have these cortical granules that are filled with enzymes. So what happens is when this sperm fertilizes this egg, there is a chemical reaction that happens, and these cortical granules release enzymes, and what happens around the zona pellucida is that it hardens. So it will not allow a polyspermy, which means it won't allow a second sperm to fertilize that one egg. Now, we see the nucleus here, although it is important to note, remember that the egg is arrested at metaphase 2, of meiosis II until it gets fertilized by the sperm. So this ovum that is traveling down the fallopian tube is not going to have a nucleus. It's going to have chromosomes arrested at metaphase 2. When this sperm fertilizes the egg, meiosis II is going to finish, and then the chromatin is going to quickly condense into a nucleus, and then it's going to allow for the fusion of the male nucleus with the female nucleus. Okay, continuing here, we have an awesome video of an unfertilized human egg. So perhaps you've wondered how in vitro fertilization is done. How can an egg taken from a female body be fertilized by sperm from a male in vitro, meaning in a petri dish? So pretty much this is what it's done. You take a really good microscope. You grab this suction cup here to kind of keep the egg from moving. And then with this micropipette, you puncture the follicular cells and the zona pellucida, and you inject the sperm. Now remember that there's going to be many sperm here, so when you inject the sperm around here, only one is going to get to actually fertilize the egg. Only one is going to make it through the zona pellucida. So let's look at the video as it plays. There's the human egg. That is not a nucleus. What do you think that is there? We'll see in just a moment as we shake the egg a little bit. So here it comes. What do you think that is? We learned about it in other videos. Yes, it's the polar body, right? Remember, the polar body is that other haploid cell that is undernourished, that is not going to develop into the ovum, right? Sometimes it just hangs out there, right, with nowhere to go. Sometimes it undergoes meiosis again and it forms another polar body. Sometimes it just gets degraded and reabsorbed into the egg or the female body. Okay, so that is it for the egg and sperm structure.